Hello everyone, welcome to the class of biotechnology. Today we are going to discuss about an enzyme ligase. This enzyme has an important role in joining the two molecules together by forming a new chemical bonds. But before we move forward, if you are new here, please like and subscribe this channel so that you cannot miss any of the future videos. DNA ligase is an enzyme that joins the two molecules together. Which type of two molecules? It can join two DNA molecules together. Two DNA together. And how it is like joining these two DNA molecules together? By forming the phosphodiester bond. By forming phosphodiester bond. For the formation of the phosphodiester bond, it requires hydroxyl group and phosphate. Suppose you are having the two DNA fragments and you want to join these two DNA fragments. This is a fragment 1 and this one is fragment 2nd. And these are having POH group and phosphate group. So in the presence of the DNA ligase, you can join these two fragments together by forming the phosphodiester bond at this position. Finally, you will get a single fragment. In this way, the formation of the phosphodiester bond in the presence of the DNA ligase ligates the two DNA fragments and represent it as a single DNA molecule. The concept of DNA ligase is first given by two scientists named Weiss and Richardson. Or you can say these two scientists actually characterized the DNA ligase in the year 1967. Which year? In the year 1967. There is a wide application of the DNA ligase. DNA ligase plays a very important role in DNA replication, recombination, and DNA repair by forming the phosphodiester bond, or you can say the catalyzing the formation of the phosphodiester bond between the two adjacent nucleotide when there is a presence of free hydroxyl and a phosphate group. It also helps to maintain the DNA integrity and the genomic stability. It helps to maintain the DNA integrity and genomic stability. For example, during the DNA replication, DNA ligase works on the Okazaki fragments to fill in the complementary bases and making the DNA continuous. Suppose this is normal double-stranded DNA. And when it got exposed to some UV radiation or any other type of modification like glycosylation or amino acid modification, the strand of the double-stranded DNA may get breaked out, or due to the modification, there is a requirement of the proofreading. So for the proofreading, there is a breakage in the strand. Here's the DNA ligase comes into account, and it again rejoins those breaked out molecule or break out the DNA strand, and hence in this way it repairs the DNA molecule. Similarly, in DNA replication, DNA ligase helps to join the Okazaki fragments of the lagging strand during the DNA replication process. Here it joins the Okazaki fragments. Another example of the DNA ligase is in preparation of the recombinant DNA. Recombinant DNA are those DNA that are created in a lab by combining the genetic material, that is DNA, from multiple sources using a technique called recombinant DNA technology. Suppose this is a plasmid vector DNA of a bacteria having a sticky ends at both the ends at 3' OH, and this one is the double-stranded DNA fragment of human, and you want to ligate these two fragments together, and this part of the human carries, suppose, having some important genes on it. So to study its structure and function, you cannot manipulate directly it into the human just because of the ethical issues, because for the study purpose you have to isolate those particular part of the DNA again and again. So to avoid the ethical issues, you can transfer this particular part into the plasmid vector of a bacteria, so that you can make a replication of the this DNA fragment into the bacterial genome. So with the help of the DNA ligase, you can produce recombinant DNA by ligating, or you can say by combining these DNA fragment, double stranded fragment of a human into the plasmid vector of a bacteria. So here DNA ligase help in the ligation of this fragment, double stranded DNA fragment of the human into the plasmid vector of bacteria. And in this way you can produce the recombinant DNA where the plasmid vector of the bacteria carry the double stranded fragment of human. Hence in this way you can easily make a multiple copies of this recombinant DNA. Recombinant DNA here carries a few part of the ligated DNA, that is the DNA from the human. This part is the part from the human, and this part is the plasmid vector of the bacteria. Hence in this way you can make multiple copies of it in bacteria cell and perform whatsoever study you want to perform. So by obtaining the recombinant DNA, finally with the help of the DNA ligase, you can easily produce insulin. It have a role in gene therapy, human growth hormone production, recombinant vaccines, enzyme production, etc. Now let us discuss in detail about the mechanism of DNA ligase. That means how it actually works, or how it participate in the formation of the phosphodiester bond. So the first step is the nucleophilic attack on the alpha-phosphorus of ATP by ligase enzyme. Actually, the DNA ligase enzymes require an energy source to catalyze the formation of the phosphodiester bond between two DNA fragments. Suppose this is the two DNA fragment and you want to combine these two DNA together by the formation of the phosphodiester bond, right? So this energy is derived from either ATP or NAD+. ATP is what? ATP is adenosine triphosphate. You can also call it as adenine-ribose triphosphate. It carries the 3 molecule of phosphate. Now suppose this is a ligase enzyme and this enzyme is carrying a lysine into it. So the oxygen atom that act as a nucleophile from a specific amino residue, usually from the lysine, within the ligase enzyme, act on the alpha-phosphorus atom of the ATP. This is the alpha-phosphorus atom of the ATP, adenosine triphosphate. This nucleophilic attack on the alpha-phosphorus of ATP by ligase results in the formation of covalent bond between the enzymes. This is the covalent bond formation here. Results into the covalent bond formation between the enzyme. Which enzyme? DNA ligase. And the AMP portion of the ATP. AMP portion means adenosine monophosphate. This is the AMP. This part is the AMP. When you remove the pyrophosphate from it, that means if you remove the two molecule or two atom of the phosphate from the ATP, it will leave behind the adenosine monophosphate. So the attack of the ligase on the ATP molecule results into the formation of the covalent bond between the ligase enzyme and the AMP portion of the ATP molecule, creating a ligase-adenylate intermediate. Simultaneously, pyrophosphate is released as a byproduct. The second step is the transfer of the AMP to the DNA molecule. So after the formation of the ligase-adenylate intermediate, or you can call it as a AMP ligase complex, when this complex comes in close proximity to the fragments of the DNA which are supposed to be joined together, then the 5' phosphate group at the end of the DNA strand, which is supposed to get ligated, this particular phosphate DNA act as a nucleophile, and it attacks on the phosphorus atom linked to the AMP molecule in the ligase-adenylate intermediate. This is a complex of what? This is a complex of ligase-adenylate intermediate. So the phosphate group that is present at the DNA strand at 5' end attacks the phosphorus atom linked to the AMP molecule in the ligase-adenylate intermediate. So this attack results into the formation of the phosphodiester bond. This nucleophilic attack on the AMP ligase complex from the 5' end of the phosphate group of the DNA molecule results into the formation of the phosphodiester bond between the 5' end of the DNA strand and the AMP molecule, or the phosphate group of the AMP molecule of the AMP ligase complex. So here would be the formation of phosphodiester bond. Phosphodiester bond will form here between the 5' end of the phosphate of the DNA strand and 5 or you can say the phosphate group of the AMP molecule in the AMP ligase complex. So after the formation of the phosphodiester bond here, simultaneously the lysine residue in the active site of the ligase is released and regenerating the enzyme in its original form. So the activated AMP molecule is transferred from the enzyme to the DNA, forming a DNA adenylate intermediate, and this intermediate is now primed for the final step of the phosphodiester bond formation between the two DNA strands. The similar phenomena will also occur at this particular strand. Here the phosphodiester bond will also form in the final step where the nick is being sealed. The 3' OH group of the nicked DNA strand act as a nucleophile and it attack on the phosphate group atom of the DNA. This particular phosphate group, it attack the 3' OH attacks on this particular phosphate group and it seals this particular nick, and this attack results into the formation of the again phosphodiester bond here will be formed. Phosphodiester bond will be formed here between the 3' OH of one DNA strand, this DNA strand, and the 5' phosphate of this particular second strand, and hence effectively sealing the nick. Simultaneously, the AMP molecule is released from the DNA, regenerating the original DNA structure. Finally, you will get the double-stranded DNA. In this way, the ligase enzyme catalyzes the formation of the final phosphodiester bond, joining the DNA strand together and hence completing the DNA repair or sealing the nick or joining the two DNA fragments together. This is the ligated DNA molecule, and this portion represents the generation of the new phosphodiester bond. Next is the type of ligase. Basically, there are three types of ligase: ligase first, second, and DNA ligase third. DNA ligase first, it is genetically modified. Actually, it is produced by genetically modified yeast, and it have a role in joining of the Okazaki fragments of the lagging strands. DNA ligase second, it is not a true ligase because it does not have its own gene and it have a role in DNA repair mechanism. Next is the DNA ligase third. It also have a role in DNA repair mechanism, specifically the nucleotide excision repair, and it is found in the nucleus. This ligase can also be found in the mitochondrial DNA. So this was all about the DNA ligase. I hope you all like this video, and if you like this, please like and subscribe, and don't forget to hit the notification bell so that you cannot miss any of the future video. So thank you so much.